for that award.
Okay, okay, okay. Good. All the best. All the best to you.
यस, तो जैसे उन्होंने बुक खरीदा था तो मुझे मैसेज किया था बुक का
हाँ।
फोटो, तो सर का
अच्छा।
बुक मैंने ऑर्डर किया,
अच्छा।
तो सर मैं भूल गया वो।
हाँ। तो आकाश शेयर का दे दिया मुझे,
यस यस सर।
तो shall I share or still time? We will wait for some more time.
अभी तो 1-2 मिनट वेट करते हैं। 2 मिनट में कोई अगर फीडबैक शेयर करता है तो वो अच्छा है, बाकी
अच्छा।
तब तक रिमाइंड।
अच्छा अच्छा, ओके। इस श्रीधर सर को बोलो ना फीडबैक देने के लिए।
हाँ यस यस, श्रीधर सर तो उन्होंने बहुत सारे वेबिनार्स अटेंड किए हैं, तो
हाँ।
सर इफ पॉसिबल प्लीज प्लीज शेयर योर फीडबैक। रोहिणी मैम आल्सो देयर, आप भी शेयर कर सकते हैं मैम।
हेलो,
वीडियो ऑन करा?
यस
हाउ
मैम।
सर? हेलो सर.
हेलो हेलो रोहिणी, हाउ आर यू?
Sir, I am very fine.
अरे
And thank you.
you
And
are
have a
back.
great evening to all.
Yeah,
And sir,
thank
thank
you.
you very much for this second webinar series. So we are able to attend this from our home because there are some. I can't say there is a problem, but the thing is that without we are able to concentrate on a single webinar when we are at home, because in a
Okay,
working place there are something or
Yeah, some
work is going on. The work is
something
going on. We can't say.
keeps coming and all that. Okay, okay.
And thank you very much, sir. I'm very blessed
Yeah,
for
thank you very
your
much.
duty. Thank you
Thank
very
you.
much, sir.
Thank you very much. Thanks a lot, Rohini. I really appreciate you are joining again to this webinar series
Because
number
uh,
2.
okay,
Yeah, that's
because sir, I'm last time also I'm saying that you are a knowledge of sea, and I want to take some drops from your knowledge.
Oh my
So
goodness.
yes, sir. So I'm always trying to catch your webinar and rebrush my knowledge which
Yeah.
I gained from your side, sir. So thank you very much, sir.
I'm overwhelmed and I feel I'm on Chana Ka Jhad.
नाही सर, नाही सर, नाही नाही सर, मला नाही वाटत आहे सर असं. नाही
I'm just
नाही.
joking.
No
No
sir,
problem.
no
No
sir.
problem. No
Actually, this
problem.
is my I'm speaking from my heart, sir. I'm not.
I understand. I really know you.
And
You
if you
don't
feel
have
I'm
to.
extremely sorry, sir.
Yeah.
मला तसं नाही
No,
म्हणायचं.
no, no. Just okay, okay, okay. Shall we start now? So,
Yes
friends,
sir. Yeah.
friends, welcome to Good Laboratory Practice webinar second series. And first series was very successful. And this is the series which we have started on demand of people, those who are interested in knowing more about GLP. So, for the first time, the second series uh, uh, is being run now. Today is the first day, and first day we will cover principles of good laboratory practice in today's webinar over there. So obviously, we will understand what is principles of good laboratory practice, how we should look into good laboratory practice principles, and what are the necessary requirements as far as GLP is concerned about that. So I will just share with my screen so that we can start with our presentation over there. Is it okay? You are seeing my PowerPoint?
Yes, sir.
Yes, sir. Yes, sir.
Okay.
Yes, sir.
Sir, yes, sir.
Okay, good. We will cover principles of GLP compliance monitoring document number 1 today. This is a first document which is very, very important, which cover most of the aspects of GLP over there. So, as you know that why we use GLP. What is the requirement of GLP? Let me give you in brief the history of GLP genesis for that matter. As you know, the drug discovery and development process. The first is early discovery, then lead optimization, then lead identification. Then we will go into preclinical studies over there. After the preclinical studies, the product will go into clinical studies. Until then, we will call it preclinical. And then when we go to human trials, that is called phase 1, phase 2, phase 3. And then it will be drug registration over there. So these are the phases of drug discovery as far as drug discovery and development is concerned over there. So how it happens, let us understand that. You know that 10,000 to 15,000 chemicals are screened for as a drug over there. So obviously out of that only 250 compounds will, that is called drug discovery program. In drug discovery program, 10,000 to 15,000 molecules will be screened. Out of that, only 250 compounds will enter into preclinical arena over there. So obviously, only 250 will come into preclinical studies. And out of that 250, only 5 compounds will enter into the clinical trials over there. That is phase 1 trial, phase 2 trial, phase 3 trials. And the amount of time required for this is 3 to 6 years for drug discovery and preclinical, and 6 to 7 years for clinical trials over there. Obviously, it's a long time over there, and then it will go to further over there as a human phase 4 trials over there. So obviously, this is called as IND, and this is called as NDA. What is IND? IND is investigational new drug over there. So what is investigational new drug? What I told you about that, 10,000 to 15,000 molecules which are discovered over there, they are called IND over there. So which are being registered with regulatory authorities over there. That 5 molecules which I said to you over there. So in exploratory studies, we have pharmacokinetic, toxicology, genotoxicity, safety pharmacology, efficacy studies. These are all exploratory studies. It doesn't require any good laboratory practice or GLP for that matter. For GLP studies, yes, the final studies which are to be submitted to regulatory authorities, they require GLP studies only. They don't accept non-GLP studies as far as registration is concerned of product. So toxicology is 4 weeks, that is 2 species. Genotoxicity, again in vivo, in vitro, both the studies. Toxicokinetics, safety pharmacology, and reproductive toxicology. This is all to be done in IND over there. Once this data is ready, then you can apply for IND submission to regulatory authorities, and you get over there, and you can go for first time in human. That is phase 1 trial over there. So obviously, NDA, this new drug application, this is what is phase 1, phase 2, phase 3, and phase 4. This is what is there that you require. Even along with that, you require preclinical toxicology, like toxicology, 3 to 6 months studies over there. Then reproductive toxicology, male fertility, and pre and postnatal developmental study, chronic toxicology, and carcinogenicity. These are studies required as far as preclinical toxicity is concerned over there before the drug is in market, coming to the market over there. So this is how drug is developed, and this requires a lot of money and a lot of time over there. So obviously it requires minimum of 12 to 15 years and minimum of $1 billion over there. So obviously because of this, the cost of drug development is on higher side over there. So only because the 90% attrition is mainly due to lack of success in translating preclinical efficacy and safety data into successful human trial. So whatever data we generate, the 10,000, 15,000 molecules over there, 90% will be dropped over there, and only 10% will go ahead for new drug development over there. So higher attrition rate, whatever efforts are put into new drug development over there, obviously because of this, the attrition rate, the cost of the drug increases over there. Why cost of drug is high? People, they ask sometimes. This is only because there is a lot of exploratory studies going on, lot of drug discovery programs are going on, so it requires a lot of money. As I told you, $1 billion over there. Obviously, because of that, the rate of drug discovery is on higher side, and success have a major impact in optimizing efficacy as well as pharmacodynamics, pharmacokinetics, and could minimal animal studies in preclinical trials after proper validation. This is how drug is developed over there. So we will not go into details of drug development. We will focus on preclinical toxicology only, because preclinical toxicology requires good laboratory practice. And why it is required, I will come to that point after maybe few slides over there. So we have to conduct extensive toxicological tests using animals to determine the drug is likely to be safe and effective in humans. So we cannot go into humans directly. Let me tell you that because of this, we need to use animals. There are alternative methods coming up. We talk about that a lot, but let me tell you that unless and until complete animal studies are undertaken, we will not be able to find out whether drug is safe and effective in humans or not. So for this, preclinical toxicology is very, very important for all of us over there. So data from all these tests should prove drug safety and effectiveness. It is not only safe, it should be effective also. So obviously, if at all it is effective and safe, then only drug will be brought to the market for that matter. These data then submitted regulatory authorities for new drug application. So obviously NDA, I told you, unless and until it is proved that drug is safe and it is effective in curing that particular disease, so obviously then only it will be approved by Food and Drug Administration over there. It is first line of defense. Preclinical study is first line of defense over there. It indicates that drug may be tested safely in humans and may be effective in curing human disease. So obviously, we are interested in curing the disease. We are not interested. We are interested, say for example, right now situation is that we are interested in curing corona infection. So obviously coronavirus is there and we need to find out a drug which is effective against coronavirus. So obviously it has to go through all these phases over there for drug development over there. It is happening over there. Every whole world is working on that. Preclinical studies help in support for human pharmacology. Obviously, preclinical studies not only give us the safe drug, but it will also help us in human pharmacology, support human toxicology, prediction of pharmacokinetics, whether it is being observed and excreted from the body or not, the pharmacokinetics, and screening of new doses, forms, and formulation. It is not only discovery, but you need to find out what doses you are supposed to take, what forms you can administer the drug into the human. That all has to be go into details of drug and development process over there. Now we will come to birth of GLP. 1 minute, I am just. So what happened in early 1970? The FDA investigated the number of cases of poor practice in toxicology laboratories throughout the USA. It doesn't happen in India. It happened in USA. So FDA inspected over there, and the result of this investigation, they near about 98 laboratories they have inspected and revealed many cases of badly managed studies over there. The studies were managed, poor training of personnel, some cases of deliberate fraud was happening. So FDA thought what should be done. So again, in addition to this, they found that poorly trained study directors, there were no study director, no trained study personnel over there, poorly designed protocols, no protocols or study plans were not there, protocols not followed, whatever protocols were there, they never followed that procedure, whatever procedure they were working with, it was not conducted as per that particular procedure. Raw data badly collected, raw data was not collected properly, not correctly identified, without traceability, not verified or approved by responsible person. There is no responsible person who can tell this study was done as per this protocol or as per this or there. They were not trained at all. And lack of standard procedures. There is no standard procedures available over there. And let me tell you that in preclinical studies, animal health is very, very important. Animal husbandry is very, very important. So there was a poor animal husbandry over there. So obviously, this all uh, was happening over there. Inadequate characterization of test atom and test system. That the reason now characterization is primary importance as far as GLP is concerned. So you need to characterize the test substance. So test atom we call it now. So test atom should be characterized over there. We must have certificate of analysis. Inadequate resources were available. Equipments not properly calibrated. That the reason we insist on proper calibration of equipments now over there in all the GLP facilities. Reports not sufficiently verified, not reflecting actual raw data, and archiving was inadequate. There is no archiving available over there. So this is the one case which I just wanted to inform you or tell you about this test facility. It is called Industrial Biotest Laboratories. It is known as IBT over there, who perform about 35 to 40% of US toxicology testing. That time it was 35 to 40% testing was done by only IBT over there. And the FDA conducted near about 867 audits of IBT. So obviously, see the beauty of that. Out of 667, 618 studies were found invalid. So obviously, the ratio is very high of invalid studies over there. Then FDA sued the IBT, and the court found IBT managers guilty of fraud. Obviously, this was guilty, and they were imprisoned. Probably, I don't know. It was stated over there. So what then FDA decided? FDA formed a task force for that matter, and the task force was formed to ensure the validity and reliability of the test data. The standards and policies enforced to evaluate the performance of research. So they thought that we must have a task force which can look into this, and they met and they discussed in lot over there what should be done as far as this type of frauds are concerned. And they decided their decision was introduce a new regulation to cover non-clinical safety studies. That's the reason it is called GLP, and GLP was born that time over there. Good laboratory practice regulation were framed over there, and draft was ready in 1976, and which was enforced in US in 1979 for that matter. So obviously you need to understand it took pretty long time for them to decide about the regulations over there for non-clinical safety studies over there. Simultaneously, in Europe, the OECD Council decision take a decision on mutual acceptance data in 1981, and they form OECD GLP for that matter. So what do you mean by that? Good laboratory practice. What it helps us? It helps us to promote quality and validity of test data. Whatever test we undertake in GLP arena, so the quality and validity of test data is very, very important for that matter. So on that basis, they have gone for GLP regulations. So what are the main goals of FDA to have this regulation? They wanted to see that whatever data or whatever results they get, it should be reliable. It can be repeatable, it can be auditable, and recognized by scientists worldwide. Let me tell you that many people, they have a notion that GLP is a managerial system. But let me tell you that science has to be done by you and me over there. Science we should not forget, and science will be looked into that by regulators over there. So obviously, we scientists, we need to do science under the GLP principles. This is very, very important over there. So what are the principles? The purpose is not to assess the intrinsic scientific value of a study. Uh, Although GLP says that they are not concerned with scientific value, but we are concerned about the scientific value of the study over there. GLP principles are set of organizational requirements. Obviously, GLP tells, because they found the frauds over there, they found the people are doing wrong things over there. That the reason they wanted that what organization should do as far as generating preclinical data in GLP situation over there. An important difference between science of a study and the organization of the study. So you must remember that this is very, very important for all of us to understand that difference is there between science of the study and the organization of the study. So we must look into that. So let us see that what GLP aim to make the incidence of false negatives. Let us understand this. I am talking to you very critical aspect of science over here. To make the incidence of false negative more obvious. That means results demonstrate non-toxicity of toxic substance. You got me? This is a very, very important aspect, and that was happening over there. So second thing is that GLP aim to make the incidence false positive. Earlier was different, more obvious, that is results demonstrate non-toxicity of a toxic substance. That is very, very important as far as when we do the science. We need to keep it in mind, what is false positive, what is false negative for that matter. So this is very, very important for us. So what GLP does then? GLP promote the mutual recognition of study data. That is what is there across the international frontiers. So whatever GLP we are doing in India, so we are not just doing the GLP studies for Indian registration. When you are GLP in India, you can submit your test data or your studies into 30, more than 34 countries in the world, including US and Japan. So obviously this is very, very important, because under that MAD treaty, under that, on the MAD treaty, US and Japan, they are also signatories over there. So we have mutual recognition of study data across the international frontiers. This is the importance of GLP over there. And good laboratory practice, as I told you, that you need to do good science with good organization. So let us understand what is good science and good organization for that matter. What is good science? You must be having your experimental design which is internationally recognized. Obviously, we have guidelines for that matter, and we need to use guidelines. The experimental designs are given over there, the based on known scientific procedure. So whatever is there, it should be based on known scientific procedure. We should control variables for that matter. Control of variable, there should be minimum variables over there, and interpretation of results should be in such a way that regulators should be accepting your data without any problem. There are no 483 letters issued to you in response to your submission to regulatory authorities. Results are acceptable to regulatory agency. That's what is very important, because what GLP does, we do the studies, but we are not aware which regulatory agency it is going over there. So obviously it should be accepted by regulatory agencies. This is what is very, very important for all of us to understand. It is for regulatory purpose, it is for product registration. So obviously when product is registered, you can market your product in the whole world for that matter. And what is good organization? So good organization, because a lot of things happened over there in FDA, they decided what is good organization. Adequate physical facilities should be there, qualified, experienced and trained staff should be available. Planning studies, that means study plan should be available. Resource allocation, that is test facility management, they have designated that, and test facility management job is to allocate the resources for conducting the studies over there. Good record keeping, documentation. We said, what is the definition of GLP? Give lots of paper. That's the only reason we say that give lots of people means good record keeping. So obviously everything should be recorded, everything should be approved by test facility management, and it should be in an organized fashion, organized manner over there. All activities should be organized in such a way that you should be conducting your study without any problems, without any issues for that matter. Implementation of verification process, compliance with GLP should be there. So what GLP does to all of us over here? So GLP limits the waste resources over there. So whenever you are GLP, waste resources are limited. Limiting use of animals. That's the reason GLP says use 5 males, 5 females. You are not supposed to use more than 5 males, 5 females for that matter. So obviously your GLP says that you must limit use of animals over there. They will not allow you to use maximum number of animals or whatever animals you feel to be used for the study. No, that is not the case in GLP. You must limit your use of animals while doing GLP studies. Ensuring high quality of results and validity of data. You must ensure your data should be of quality and results should be valid for that matter. And ensuring comparability of results. So obviously you must be able to compare your results with international results available over there, and you must be able to say that yes, your product falls in line with international arena. So promote mutual acceptance. I told you about this mutual acceptance of data, in that your data can be submitted to any country over there. So GLP is a managerial concept for organization of the studies. That is what is there. I can summarize over here that GLP is a managerial concept for the organization of study. You must organize the study in a better way, in a quality management system over there. So GLP documents, there are a number of documents we are discussing today, document number 1, but there are a number of documents for your information. I'm just highlighting few documents for your information. These are all available on NGCMA website. You can also download them and you can go through them very easily. Document number 1 tells us principles of GLP and compliance monitoring, which we are going to discuss in today's webinar over there. Document number 4 will tell you about the quality assurance and GLP over there. And document number 5 will tell you compliance of laboratory suppliers with principles, GLP principles over there. Even suppliers, whatever suppliers, there are animal suppliers, there are chemical suppliers, there are instrument suppliers. There is a document for that. You just cannot have your suppliers just for your own interest. No, that is not the case in GLP. Suppliers are also having some responsibilities as far as GLP is concerned. So obviously, there is a document for that also. Document number 6 will tell you about the application of GLP principles, field studies. Yes, you can do field studies as well. There are some organization, they are doing field studies. Even in Pune, there is one organization, they are conducting field studies as per GLP. So obviously, field studies, how you can conduct field studies, you can refer this document number 6 for that matter. Document number 7 is application of GLP principle to short-term studies. All acute studies or ecotox studies or genotox studies are called short-term studies, and you can refer your document number 7 for that. And document number 8 is very, very important because it tells you about the roles and responsibilities of the study director in GLP studies. So obviously, study director is very, very important as far as GLP is concerned, and it tells you about the roles and responsibilities of study director. There is a document number 8 for that matter. Document number 11 tells you the roles and responsibilities of the sponsor in application of the principles of GLP. And document number 13 will tell you about application of OECD principles of GLP organization, management of multi-site studies. My colleague is going to talk about this multi-site studies. Jyoti will be taking up this. All these documents will be discussed in our webinars. Obviously, you don't have to worry about the documents over there. We are covering almost most of the documents which are given in GLP documents over there. Document number 14, the application of GLP principles in vitro studies, how you can conduct in vitro studies, that also is document number 14 over there. Establishment and control of archiving that operates compliance with GLP. Finally, archiving is the most important aspect of GLP, and it tells you about how to operate your GLP archiving in your facility. Obviously, this document is also available for you, your reference. Now, what is the scope of GLP? Yes, we have talked about these documents over there. Let us see what are the scopes and where you can apply your GLP over there. The pharmaceuticals, that means drugs over there, you can, if at all you drug discovery program, as I told you about this. Agrochemicals for that matter, because agrochemicals are very, very important because they are dealing with sites, pesticides. Obviously, safety is very, very important as far as agrochemicals are concerned. Cosmetics, yes, cosmetics are also very, very important because we apply cosmetics on our body every day. Even simple your skin irritation and sense sensitization studies are undertaken on cosmetics. So we must find out whatever cosmetics we are using in day-to-day life, they are free from all this and they are safe for use over there. Veterinary drugs, obviously we are not aware of how many veterinary drugs are coming into market. Obviously, veterinary drugs are also undergoing GLP studies over there. Food and field additives, and now they have added medical devices. So far, medical devices were not included in scope of GLP, but this year, probably 2020, they have added medical devices into scope of GLP over there. Even if at all medical devices testing is done under the supervision of good laboratory practice. These chemicals may be synthetic, may be natural, or may be biological origin for that matter. So what is the definition of GLP for that matter? As I told you, GLP is a quality system concerned with the organizational process and condition under which non-clinical safety studies are planned. That means you are supposed to plan your studies. That means there should be a study plan available over there. And as per study plan, as per your plan, you should be performing your study over there. That the reason performing, conducting of study is as per your study plan. That is perform. Third thing is monitor. There should be somebody who can monitor your studies over there. That means you must have quality assurance over there in your facility over there. And whatever you perform, you should be able to record that legibly, promptly, there and there only. That the reason recording has to be promptly, legibly there. Animals cannot wait for their symptoms. Okay, so obviously when you dose your animals, you should be waiting for the symptoms to be observed over there, and you should record symptoms there and there only. This is very, very important as far as animal studies are concerned, because they are in captivity. They won't tell you, they can't speak to you. Obviously you need to observe animal very nicely, very effectively. Observation should be there. And finally, whatever you monitor, whatever you record should be reported over there. So and whatever is reported should be archived. It should be available in your archiving. This is the definition of good laboratory practice. Very, very important to understand and implement in your GLP facility over there. There are some definition. I will not go through most of the definition, but for your information, I will just read what are the definition over there. Test facility over there. What is test facility? Because many times we say test facility, test facility. You must understand what is test facility. Means the person premises that are necessary for conducting the study. You also hear about test facility management. Yes, you have test facility. There should be test facility management. The person who has the authority and responsibility for the organization of its functioning. That is test facility management. Maybe you have deputy test facility management, maybe you have one or two test facility management, no problem over there. But there should be somebody designated test facility management over there. And sponsor, who is sponsoring or who is placing the studies with your facility, he or she is called, the organization is called sponsor over there, an entity which commissions a study to your facility over there. And as I told you that study director is an individual responsible for overall conduct of the study over there. The study director is very, very important person as far as GLP is concerned. And quality assurance, yes, there should be somebody who can police you over there. Unless and until police is there, you traffic moves properly. If there is no police, there is no quality assurance, traffic will be jammed over there. As you know that traffic will not move properly. So that's the reason GLP said, yes, there should be a quality assurance in the facility who can monitor the studies as per GLP principles. Now there are few more definition over there. Study plan, as I told you that your planning should be there, a document which defines the objectives and experimental design for the conduct of study. It should be ready before study is initiated over there. And there are things, yes, GLP is also taking into account if at all there are any planned change in your study plan. So it is called amendment. An intended change to the study plan is called amendment, and unintended change from study is called deviation. Yes, you are allowed to do that, but it can be amended, it can be deviated unless and until you report that whatever has happened in your study over there. It is very, very important. And there are some other additional definition like test systems, any biological, chemical, physical system used in the study, raw data, again over there, all original test facility record and documents which are results of original observation and activities in the study is called raw data. You must design, you must define what is your raw data. There are instruments which are giving thermal printouts in your facility. Obviously, you need to find out whether they stay for how long, maybe 1 month, 2 months. Maybe you have printouts over there and you get direct printouts attached to your instruments. So obviously you need to define what is your raw data over there and which is your original test facility record. You must define in your standard operating procedure over there. These are very, very important definition. You must keep it in mind. Experimental starting date, experimental completion date, study initiation date, and study completion date. Experimental starting date, the date on which the first study specific data are collected. That means first time whatever you record, that will be your experimental starting date. Once you get the animals into your room, you acclimatize them. When you acclimatize them, you start recording the health aspect of animal. So obviously, when you record something, the first time when you record, that is called experimental starting date for your experiment over there. Experimental completion date means last date on which data are collected from the study. The last observation when you record in your study file, that is called experimental completion date. And very interestingly, there are two other additional definitions which are very, very important as far as you and me are concerned. Study initiation date. This is the date on which study director signs on study plan. So when you prepare study plan and study director signs, that is called study initiation date. And study completion date is the date the study director signs the final report. This is very, very important. That means your final report is ready and now study is closed. So obviously these are definitions, specimens, test item, reference item, batch, vehicle. These are all additional. Specimens are from any material derived from the test system for examination, analysis, retention. Test article is the subject of the study because of test item we are all doing GLP studies. That is the reason test item is also again very, very important. Reference item means we use test item for comparison. We get control item or reference item over there. So obviously any article used to provide basis of comparison as far as test atom is concerned. We must find out whether it is behaving as per your product in the market or not. So obviously, the reference atom is used in many GLP studies. Batch is a specified quantity or lot of test atom produced during the defined cycle of manufacture. Vehicle, any agent which serves as a carrier to mix, disperse, or solubilize the test atom is called vehicle over there. So obviously, these definitions are very, very important as far as you and me are concerned. So obviously, looking into good laboratory practice, the document number 1, principles of good laboratory practice, I have formed the 10 commandments of that. Good laboratory practice principles is covered under the 10 commandments. If you look into all these 10 commandments, you are following GLP over there. So first commandment is test facility, organization, and personnel. Second commandment is quality assurance program. You must have quality assurance over there. And third commandment is test facilities over there. Whatever test facilities are there, you must have that facility suitable and good construction over there. Fourth is apparatus. We use apparatus, we use instruments, equipments. So we use materials like chemicals and all this, reagents and solutions over there. So for that, we must, this is fourth commandment for that matter. Fifth commandment will tell you about the test systems. Whatever test systems we are using for GLP studies, they will be covered under that. Test and reference material. Obviously, now we talked about test atom, test and reference material, the sixth commandment for that matter. And seventh commandment is standard operating procedure. In GLP facility, nothing should move unless and until you have standard operating procedure. All the activities should be covered under standard operating procedure. No activity should be performed in GLP situation without standard operating procedure. This is how you must see to that. So test facility, let us see test facility organization and key personnel over there. There are 4 key personnel as far as test facility organization is personnel is concerned. Sorry. Test facility management, study director, principal investigator, if at all you are having multi-site study over there, study personnel, and most importantly, the archivist. Archivist is considered to be the key personnel as far as GLP is concerned over there. What is test facility management? Test facility management means the person or person who has the authority and formal responsibility of organization functioning the test facility according to principles of laboratory practice. Test facility management has to provide all resources over there. The functioning of all organizations should be governed by test facility management. We should look into all in totality what is happening into test facility over there as far as GLP is concerned over there. The compliance of GLP principle, that is one of the most important aspect of test facility management responsibilities over there. And he should also look into that test facility has qualified personnel, appropriate facilities, equipments, and materials are available over there. The maintain records of qualification, training, experience, and job description. I will, because somebody is going to cover this aspect also. Qualification, training, experience, and job description are very, very important as far as GLP is concerned. You must define. There is no guideline available in GLP as far as qualification, training, and experience is concerned. But you must define in your SOP what qualification people are required for conducting particular studies you are conducting in your facilities over there. So this is very, very important over there. Ensure that personnel clearly understand the function they are performing over there, and necessarily provide training over there. This is the test facility management. If at all they don't understand, you need to provide them a training over there. And appropriate technically valid SOPs are established and followed. This is what there I told you about this. You must have all activities should be covered under SOPs over there. You must have appropriate and technically valid. That is what is very important. Don't forget that. I am emphasizing on that. Appropriate and technically valid SOP. For conduct of study, you must be looking into what are the techniques, what are the methods you are using for conducting that particular study. So obviously you need to look into the science of study over there. Maintain historical file of all SOPs over there. Ensure there is a quality assurance program. I told you that quality assurance should be there. And sign the study plan amendment and maintain over there, because in India or in most of the GLP facilities, test facility management is supposed to sign on study plan, sign on amendments, and maintain copies of this over there. So this is very, very important as per a test facility. Each study ensure that sufficient number of personnel is available. This is what is there, because in present situation we are facing some issues over there, but this is very, very important. Sufficient number of people should be available in a timely manner. See, obviously GLP tells you that people should be available, but in a timely manner. When you are supposed to dose the animal, that time dosing people should be available over there. Observation should be taken over there, obviously in a timely manner, because animals are not going to wait for you for that matter. And designate study director if necessary and replace the study director. You must find out who can be study director. You must have a SOP to designate somebody as study director over there. And in case there are some problem, there are some issues, or study director may uh, leave your organization, you must replace study director for that matter. Or he falls sick over there, or he or she falls sick, you must replace the study director for that. Responsible for management, or there should be somebody who is responsible for archiving. Test facility management responsibility is also to maintain a master schedule over there, ensure that test facility supplies meet requirements. As I told you, the suppliers are also very, very important as far as GLP is concerned, and you must see to that supplies meet requirements. Whatever instruments you are buying, it should meet your requirement. It should be for intended purpose only. Ensure that test and reference items are appropriately characterized. Unless and until it is not characterized, it will be very, very difficult to prove that what test item you have worked with. So obviously it should be properly characterized. That's the reason most of the facilities, they ask from the sponsor the certificate of analysis from the sponsor over there. You must at least have certificate of analysis, so that you need not have to characterize the test system, test item in your facility. Ensure that computer systems are suitable and validated. Nowadays, yes, GLP is putting more emphasis on using computers for data entry, or because we are all habituated now using computers for most of the things which are undertaking under GLP condition, even data entry or data calculation, whatever is there. So obviously we are no computer, we are unable to work over there. Obviously in that situation, they want to see that the computer systems are suitable for use, and it should be validated. There should not be cut and paste system available in your facility. That gives lot of problem, lot of issues. GLP inspectors found, because of that, they say that you must be have dedicated computer system for GLP work. You must have that. Ensure that clear line of communication exists between study director and quality assurance. I know that this is very, very difficult. Study directors always have a conflict of interest with quality assurance, or quality assurance have conflict of interest with study director. So state facility management responsibility to see to that good communication exists between study director and quality assurance. In the event of multi-site study, appoint principal investigator and evaluate replacement of principal investigator. It is like study director only. You must be appointing principal investigator and replace the principal investigator. Now coming to study director responsibility, we have seen so far test facility management responsibilities. Now let us see what are the study director responsibilities are there for that matter. He is overall responsible for conduct of study over there. He is a single point control. That is the reason it is said that in guideline it's said that overall conduct of the study as a single point control. This is very, very important as point, one should be noting that. So single point control, he is the only single point control. Of course, singly he cannot handle complete study. There are support systems available, study personnel are available over there. So obviously, but study director is very, very important. He should be managing the study over there. Approve study plan and amendments by dated signature, that is study director's responsibility. Ensure that quality assurance has a copy of study plan and communicate effectively during the conduct of study. Unless and until study plan is available with quality assurance cannot plan their audit or cannot plan their inspection accordingly over there. So obviously quality assurance should have a study plan over there, even amendments over there. Ensure study plan amendment SOPs are available to study plan. This is not only quality assurance, but study plan and amendments should also be available. And SOPs, in addition to this, SOPs are available to study personnel. Define the roles of principal investigator in case of multi-study. Who will do that? Study director, because study director is a single point control over there for the study. Ensure that procedures specified in study plan are followed. Yes, study plan should be ready. Study plan should describe the procedure, what procedure you are using, what method you are using for your conducting study. You must ensure that this procedure is followed by all the people over there. Ensure that all raw data generated are fully documented, recorded. Raw data is very, very important, because raw data, unless and until raw data is generated properly, legibly, promptly over there, and documented properly, you will not have proper raw data over there. So you must define what is your raw data and how you are documenting your raw data in your files. Ensure computer system used, I already covered this point. Sign and date the final report to indicate acceptance of responsibility of GLP compliance over there. So finally, final report has to be signed and dated by study director and giving a statement of GLP compliance. He has conducted or she has conducted the study as per GLP compliance only, OECD GLP compliance. Ensure after completion of study, what he is supposed to do after completion of study plan, final report, raw data, and supporting materials are archived. Archiving is very, very important. They should do that. Study plan amendments and relevant SOPs are available to study personnel. For multi-site study, defined roles of principal investigator. Procedures specified in study plan are followed. This is what is study director's responsibility. Unless and until study plan is followed, there is no issue of having study plan for that matter. When you have study plan, you should work as per study plan. And who will look into this? Study director should look into this, whether people are working as per study plan. Can somebody mute everyone over there, because I am getting disturbances? Okay. Can somebody mute everybody over there and so if there are any deviation in study, he needs to acknowledge that to study during the conduct. All raw data generated are fully documented and recorded. Computer system used in the study have been validated. These are repeatedly, I am just referring only because you must understand the importance of computerized system over there and validation of that computerized system, because every now and then your computer systems are getting updated and upgraded over there. So you must understand what computer system you are supposed to use in your GLP studies over there. Sign off the final report. That's what I told you, that finally study director has to sign on that final report. And archiving of study plan, final report, raw data over there, materials over there in the file. Similarly, principal investigator responsibilities is same as study directors over there. Obviously, delegated phases of study conducted in accordance with GLP principles. So obviously, principal investigator responsibilities are similar to study director, but only for that part of study over there, because it is not going to be full study, because the principal investigator will be conducting the part of that particular study over there. Now, study personal responsibility must be knowledgeable in principle of GLP, because you must give training to study personnel as well. They must know what is GLP is all about. What I am talking to you now, even study personnel should be also aware of this over there. And will have access to study plan, appropriate standard operating procedure, any deviation from study plan, because deviation can be recorded by study personnel over there, because study director will not be available every time to the study over there. Obviously, study personnel will be there, and the study personnel, if at all they observe any deviation, say for example, you have written in your study plan that temperature of the room will be 22 plus or minus 3 degrees, and when study personnel will enter into the room and he will find the temperature of the room is 26 degrees centigrade, obviously it is deviation, because you can't control that. So obviously you need to record that and as a deviation in your study file. So but it should be communicated to study director or principal investigator, and finally study director will be approving that and justify the impact of that particular thing on the study. All study personnel are responsible for recording raw data promptly, legibly, and should be aware of health precaution, because health is very important. All these efforts what we are putting into conducting preclinical studies or toxicological studies, we call it safety studies over there. So obviously when we call safety to the human being, we are all human beings, and whatever facilities, whatever we talk about, that it is not only for animals, it is also for human beings or people those who are working in the facility over there. So there should be temperature control over there. Animals are there, you must change all this is only because we are working in animal facility over there, so we must take care of that. And when you have any issues with the health, you must minimize the risk to themselves, to yourself, as well as the animals as over there. So obviously you need to have health precautions for study personnel over there. Now coming to second commandment is quality assurance program. Uh, it is very important, as I told you about this. When there is no police, you know that what is happening on Char Rasta over there, on crossroads over there, obviously traffic will jam over there. So quality assurance is very important as far as GLP is concerned, and they must be monitoring the studies as per OECD GLP principle. They are not only representing test facility management, let me tell you that they are also representing National GLP Compliance Monitoring Authority, Government of India over there. So obviously they are working over there to ensure that the test facility is working as per OECD GLP principles or not. So this is very important aspect of quality assurance program. You must have a documented quality assurance program. What I said to you, you must write down that, and you must keep that program available to view by GLP inspectors when they are visiting to your facility over there. So responsibility of quality assurance personnel include that maintain copies of all approved study plan and standard operating procedures with them, because they must know what is happening in study, or they must know what SOPs they are referring to while doing or while conducting study inspections, study audits over there. And should have access to master schedule. Yes, master schedule should be there, and the master schedule is giving all details of studies conducted in the facility over there. So obviously master schedule should be kept ready. Every month it should be published in your facility, and you must have master schedule of this whatever studies you are conducting, GLP studies or you are conducting non-GLP studies. It should be reflected in your master schedule. You must give that this study is non-GLP, this study is GLP over there. So obviously all studies whatever you conduct, because based on this GLP inspectors will evaluate the load of your facility and how many studies you are conducting in your facility over there. Verify study plan as per the principles of GLP. Verification should be documented. Not only verified, it should be documented as well. The conduct inspection which are study based, facility based, and process based over there. The inspect the final report to confirm that methods procedures and observations are accurately, completely reflect raw data of the study, because study quality assurance should ensure that whatever raw data they're referring in some short-term studies, they can also have 100% audit of your study or raw data, and see that it is accurately and completely reflecting the raw data over there, because that is their major role as far as quality assurance is concerned. And promptly report any inspection results in writing to management and study director. If at all they see something over there, something is going wrong, or they should record that and immediately inform to the management and study director. This is very, very important, and management should also take into consideration whatever quality assurance is saying, and try to have action taken report from the study director. Prepare and sign a statement to be included in final report. Obviously, study director has to give compliance statement in final report. Similarly, quality assurance should also give a statement that at what point of time they have audited, what point of time they have inspected the study as far as the particular GLP study is concerned, and that should be included in final report over there. Again, to just to conduct inspection and audits over there, I told you study based, facility based, process based. Maintain the records of inspection and audits. Draft report audit, because this is very, very important. All draft reports should be audited over there, and check and verify results accurately and completely reflect raw data. This is again over there. Important components of GLP. Now we are covered the quality assurance point, and now we will go into further commandments over there, uh, like facilities, apparatus and materials and reagent, test system, test and reference item, and standard operating procedures over there. Now, third commandment is facilities. Test facilities should be of suitable size. This is what is said in the document number 1. Of suitable size. They have not given what size should be there. So you need to decide what suitable size is all about. So what is construction? What location? Adequate degree of separation, activities to assure that proper conduct of study. This is all given in the document number 1, but you need to decide what is suitable size. If at all you are using rat and mice, what is the suitable size for that? How many number of rat and mice you are going to house into your room over there, and what construction you should be constructing your facility, where it should be located, degree of separation, whatever activities you are performing, there should be degree of separation for that matter over there, and enough rooms to assure that isolation of test system, individual projects and it. You cannot conduct two spaces in one room. Rat and mice cannot be studies cannot be performed in one room. Obviously, you need to have different rooms for conducting different, because you need to have isolation of test systems, individual products over there. So obviously, if at all you have any separation in your facility to conduct the study, you can also conduct two studies, two rat studies in one, oral and dermal. Yes, you can do that, but for that you must have identification, animal identification systems available for that matter, and there should be a separation between acute study and dermal study over there. So you need to find out what is required for you and how many number of studies you conduct. Based on this, you need to find out what is that you can have in your facility. Suitable area should be available for the diagnosis, treatment, and control of disease of the test animal. Yes, animals are there. Like we are all human beings, we tend to fall sick. Obviously, animals are tend to fall sick. You must find out what is the what is wrong with the animals, and if at all it is wrong with the animal, you need to have diagnosis of that. You must be able to give treatment to them, and whatever you find, supposing there are, you are working with rabbits, and generally I have seen there are a lot of issues with rabbit facilities over there, and a lot of diseases over there. So how to control that? Your test system, when you say, when you use your GLP study animal, your facility or animal should be free from any diseases over there. Obviously, for diagnosis, treatment, and control, there should be a room available, suitable area should be available. And apart from size and construction location, minimum disturbance, separation between activities like test system facility, test and reference atom, you cannot use two test atom in one room over there. So obviously one test atom only you can use for oral and dermal, for that matter I told you. Two test systems, test atom you cannot use in one room. This is very, very important. You must have separation for archiving, waste disposal, and pest control. You must have facilities for this. Test system facilities should be designated to minimize effects on environmental variable. That's the reason we said your environmental control should be available in test system animal facilities, for that matter. Uh, the encountering disease, as I told you that you must have disease control over there, or you must find out what diseases are prevailing in your facility. Encountering other test items, sufficient rooms to ensure isolation of spaces and project suitable for room for diagnostic treatment and control. Separate areas for storage of supplies. You must have separate areas for field and water storage over there. Test atom facility. So what is test atom? It is subject of study over there, as I told you that, and it is very, very important over there. And you must avoid mix up, and you must avoid contamination. You must have separate areas for receipt of test atom facility, storage facility, weighing facility, mixing of formulation preparation, dispensing of test atom, and preserve identity, concentration, purity, and stability of test system. You must have this for test atom facility over there. You must have archival facility, because archiving is very, very important. You must have archival facility, designated area for facility for the secure storage of retrieval of study plan, raw data, final reports, samples of test system specimens. They are all there in archival. So you must be able to have a archivist, designated archivist for that matter. Design and condition based on protect materials from untimely deterioration. You must see to that whatever materials, whatever raw data, whatever study plans, whatever things are available in archiving, they should be not deteriorating over there. They should be safe in your archiving. Nowadays, lot of emphasis is given on waste disposal facility by GLP inspectors, because handling and waste disposal procedures should not jeopardize integrity of studies. This is what is there. And for waste, you must have collection points, where you are storing, where you are disposing, what decontamination you are using into this, and how you are transporting your waste from the facility to waste disposal facility over there. So obviously this has to be looked into when you are handling waste in your facility over there. Facilities or feed and bedding. I told you that you must have feed and bedding facilities, for that matter, with adequate protection, infestation, archiving facility. I told you about the handling of disposal waste. I already told you about this, because this has to look into when you have feed and bedding, because feed is there. So obviously this feed is for your animals. That means you must have external animals coming to your place over there. You must have pest control available into your that. Now the fourth commandment is apparatus, materials, and reagent. Apparatus used in study should be clean and calibrated according to SOP. Apparatus and materials used in study should not interfere with the test system. Over there, I am just going fast because it is already 8:10 over there. Use of computer system program should be validated. Materials and reagent is very, very important, as I told you, because whatever chemicals you are using, reagents you are using, solution you are using should be of high standards over there. Because I share with you our experience with chemicals and reagents. Our purchase officer, he thought that these are cheap chemicals and cheap reagents we will buy from that, and he bought that. But unfortunately these chemicals and reagents were not suitable for your histopathology work. So obviously you are doing science, and for doing science you must have chemicals of and reagents of standard grade over there. So you cannot compromise on this. You must be looking into this materials and reagents over there. What is the expiry date? What is the specific storage instruction? What is the source of this over there? And preparation date, preparation date, spelling mistake over here. Expiry date can be extended based on documented evaluation of analysis. You can extend your expiry date, but you need to have a analysis done of your solutions or reagents and chemicals for that matter. Coming to apparatus, yes, we are using a lot of instruments for conducting the studies over there. They should be of suitably located, appropriate design and capacity. You must have SOPs and logbooks for all equipment, calibration, cleaning, preventive maintenance schedule, calibration traceable to national and international standard. Use validated equipment and computerized systems over there. This is what is as far as coming to fifth commandment is test system over there, physical and chemical over there. When you are working with chemistry, your instruments are your test systems, or your chemicals are test systems over there. So biological test system, proper conditions should be established and maintained for storage, housing, handling, care of biology, because we are using animals like rat, mice, rabbits, guinea pigs. So obviously you need to have proper conditions to establish and maintain storage and handling over there. Newly received test system should have a quarantine unit on your facility over there, and their health status should be evaluated. At the experimental starting date of the study, test system should be free of any disease. That's what I told you about that. You must ensure that when you start the experiment, your animal should be free from any diseases, condition that might interfere with the purpose and conduct of study. So you must look into that, and you must assure that whatever animals you are using, whatever animals you are, they are free from any diseases. Record of source, date of arrival, condition of test system should be purchased, because when you purchase from outside source, you must record what source you are buying, what is the date of arrival, and what is the condition of test system should be maintained. Test system should be acclimatized, quarantine first, and then acclimatize over there for the environment. All information, identity of test system should appear in their housing condition, because animal identification is very, very important in test systems as far as safety studies are concerned, or toxicity studies are concerned. So you must have animal identification system well in place in your facility. During the housing container, test system should be clean at appropriate interval. Yes, animal husbandry, again here we are talking about, and animal husbandry should be very, very up to date, neat and clean over there. And medium for test system should be changed as required. Obviously, use of pest control agent should be documented, because as I told you that pest control has to be there, and lot of emphasis GLP inspectors are putting on pest control you are conducting your facility. Now coming to sixth commandment is test and reference items, receipt, handling, sampling of storage, because test item is very, very important. As I told you that this is the subject of the studies, and you must have all the records, including test item, reference item, characterization, date of receipt, date of expiry, quantities received, because this is very, very important. This they will check, GLP inspectors will check what quantity you have received and what quantity you have used for studies, and what is left out in your facility over there. They will measure if at all they have any doubts over there, and used in studies should be maintained. And I have seen that GLP inspectors, they do it over there. And handling, sampling and storage.
ऐसी क्या बात है बता नहीं रहे हो
Hello. Storage container should be carry identification and expiry date specific over there. Test and reference, as I told you, the characterization is very, very important, and you must do characterization. If you are not doing, you ask your sponsor to give you a certificate of analysis for that matter. Each test reference item should be appropriately identified with code, chemical abstract, service number, cache number, name, biological parameters. If at all you have any information available on that particular test stream, you must record that. So you must have that, including batch number, purity, composition, concentration, test reference item. All this has to be there. In case where test item is supplied by sponsor, there should be mechanism developed in cooperation with sponsor and the test facility to verify identity of test item subject to the study over there. So obviously, when the sponsor is sending, you can have, or you can devise, you can prepare a format for test item information from the sponsor over there, and you can ask for the details from the sponsor over there. the stability over there and reference under storage condition, whether it is stable or not stable under storage condition, what should be the storage condition, you must ask to the sponsor. If the test has been administered or applied, what vehicle you are using, that should be there. And when you are formulating that particular test atom, what is the homogeneity, what is the stability, and what is the concentration of test in that vehicle, you must determine that beforehand over there. When you say that you are dosing the animals 100 ppm, so whether it is 100 ppm or 98 ppm or 102 ppm, you must know that. And because the toxicity may vary between this over there, uh, uh, maybe it's milligrams and nanograms, toxicity may vary. A sample from analytical purpose of each batch should be archived over the studies over there, except this thing. Now coming to seventh commandment, standard operating procedures. A test facility should have written standard operating procedures over there. As I told you that you must have all the activities covered under the standard operating procedure. Each test facility unit area should have immediately available. Not only have, it should be immediately available over there, and relevant SOPs and activities being performed. GLP inspectors will look into this. If at all you are having some activity being performed in your facility, when GLP inspectors are visiting, they will check whether you have SOP for that activity or not. So obviously this is very, very important aspect of GLP. You must look into this, and all the SOPs should be available when you perform that particular assay. Deviations from SOP related to study should be documented. So study plan, we discussed about deviations over there, but deviation from SOP, yes, you can have deviation from SOP. It is not that GLP gives you a chance to have or deviate from your SOP, but it should be recorded, it should be documented. And if at all it is deviated from 1 time, 2 time, 3 times, it's okay, but 4th time it should not be deviated, then you must revise your SOP for that matter. Let us see what type of standard operating procedure you are supposed to have in GLP facility. Test, reference item, receipt, labeling, handling, identification, sampling and storage, apparatus, use, maintenance, cleaning and calibration, materials, reagent and solution, preparation and label. This is very, very important. I give more emphasis on preparation of reagents and solution, because this is has to be recorded. How many milligram or how many gram you have taken the reagent, and how much solution you have prepared, what solution you have prepared, how much you have prepared, whether you have prepared serial dilution or not, that has to be there. And all the reagents or solution you prepare has to be properly labeled over there. Computerized system, yes, validation, operation, maintenance, record keeping. As I told you, raw data, what you need to define that, what is that, storage and retrieval, test system, room preparation, environmental room condition, test system, procedure for receipt transfer, proper placement, characters, identification, care of test system. You must have standard operating procedure for this. Test system preparation, this is very, very important. Observation, what examination, what observation you are going to conduct on animals, you must have beforehand. What examination you are going to do with the observation, you must have before or during the study. You must be having standard operating procedure for that. Handling of test system, individuals found moribund or dead. Yes, some animals you can find dead over there after dosing, or maybe before dosing, maybe because of they are having some disease over there. You can find that they are moribund. You must have some standard operating procedure how to handle this situation over there. Collection and identification, handling of specimen, including necropsy and histopathology over there. Standard operating procedure, as far as quality assurance is concerned, they must have their separate standard operating procedure for their planning, their scheduling, their performing, documenting, and reporting inspection. They must have separate standard operating procedure for that. Now coming to performance of the study over there. Now we have 8th commandment, which is very, very important. Finally, we have come to conducting the study over there, and performance of study, how we can perform the study. For each study, you must have a written study plan. As I told you over there, you must have a study plan before the study is initiated over there, and should exist prior to the initiation of study. The study plan should be approved and dated by study director, because as I told you, this is the study initiation date when study director signs on study plan over there, and it should be verified by quality assurance for GLP compliance. Unless and until it is qualified and quality assurance is verifying that, you are not supposed to have study plan and date and signature on that over there. Amendment to study plan should be justified and approved by study director. Deviation from study plan should be described, explained, signed and dated by a study director over there. So these are very, very important. You have amendments, you have deviations over there. You must justify that whatever amendments you are making for study plan, whatever deviation you are having in study plan or SOP, you must record that in your while performing your GLP study. And there are some important events of the study. I just wanted to highlight that: study plan preparation, study initiation, that study signs the study plan, conducting the study, experimental start date. I have given you definition for that matter. First draw data general, experimental completion date I have given. Study report preparation over there. So obviously when the experiment is completed, you must prepare your report, and these are events of the study. And study completion, when you final report is ready and study director signs over there. And finally, everything should go into your archiving over there. I already explained to you study plan. You have amendments and deviations over there. And it is a planned change to the study plan, that is amendment, and unplanned change or unintended change for study plan is called deviation over there. And you must have impact evaluation for both things. You must justify what are the planned change, and you must evaluate what is the impact of this. I told you temperature has gone to 26 degree. So you must evaluate the impact of that, whether it is going to impact on the study or animals over there. So based on this, you must give that impact evaluation in your report over there. Conduct of the study, you must have a unique identification of the study. Every study should be uniquely identified in the facility by study number, study director number, or sponsor number. The all specimen samples should carry this identification over there. Study should be conducted in accordance with study plan, and data should be recorded directly, promptly, accurately, legibly. That's what I told you, that when you record your data, should be recorded directly, promptly, accurately, legibly, and I add here there and there only. Obviously, this is very, very important. There and there only is very important. Entry should be signed and dated. Whoever is recording promptly, legibly, he or she should be signing and initialing over there with date over there. Any change in raw data without obscuring the previous entry, indicating reasons for change with dated signature over there. Now we have come to ninth commandment, that reporting of study results over there. So we have conducted the study over there. Now we are preparing the final report over there. So a final report should be prepared for each study. This is what is there. So obviously final report has to be prepared by study director. Reports of PI or scientists involved, supposing scientists, why I said scientists over there, histopathology or biochemist, you must be getting reports from them. Obviously their report will also be appended to your report. Principal investigator, obviously, yes, you are subcontracting the activity over there. So obviously that report along with the scientist report, you must be, they must be signing on dating over there. Date has to be put on this reports over there. The final report should be signed and dated by study director over there. Finally, the study end date, the study completion date is final report signed by study director. Correction and addition of final report should be in the form of amendment, signed and dated by study director. Obviously, once study director signs on final report, it is called final report only. So obviously, when you want to have any modification, any correction in your final report, obviously that will be considered as amendment to the final report. It is called amended final report only. So obviously before this, you must think about what final report you are making and how you are finalizing that over there. Finally, reporting of the study results over the content of the final report, identification of study, test and reference item, dates of experimental start and completion date. You must give that in your final report. Quality assurance statement, description of test materials and test methods over there, results and conclusion, and storage location, because you need to give in your final report where you are uh, archiving your all data, all study plan, all raw data, specimens, materials over there into your facility over there. There is a rules of ones over there. So obviously you must consider the rule of ones. There is a one study plan. One study plan, one study director, and one compliance claim over there. You cannot have OECD GLP means OECD GLP. You cannot have FDA for that matter. When you say FDA, it should be only FDA for that matter. And one final report. There will be one final report for that matter. There will not be two final reports. As I told you, if at all you want to have, you can have amended final report for that matter. Now the 10th commandment is your storage and retention of records and material. This is again, I told you, archiving is very, very important. Now over there, the following should be written in archiving. Whatever we have covered so far, all 10 commandments, the study plan, raw data, samples of test and reference item, specimens, and the final report of each study should be archived in your facility over there. So records of all inspection, quality assurance will do perform quality assurance audits or quality assurance inspection, and will as well as master schedule should also be archived in your archiving over there. Records of qualification, training, experience, and job description of personnel should be archived in your facility over there. They should be available whether you stay there or you don't stay there. But when you conduct the study, if at all by chance GLP inspectors will select your study when you are available into that facility. They will see your qualification, training, experience while you were there in the facility. Records and reports of the maintenance of calibration, validation, documentation of computer systems over there. The historical file of standard operating procedure. ISO standard operating procedure is a dynamic system over there. You must be revising, reaffirming standard operating procedure. You must be selecting other test procedures over there. You must revise your standard operating procedure. So all historical files should be available into your archiving. Environmental monitoring record. These are very, very important. Every room should be monitored over there. Whatever, what is the temperature, what is the relative humidity over there. All these records should be available into your archiving. Materials, every room over there, I'm just telling over there. Materials written in the archiving should be indexed so as to facilitate orderly storage over there. And archiving is a technique, archiving is an art. You must have indexing over there, and you must as per indexing you must archive your all documents, all specimens, all records for that matter. And in archiving, only authorized person will be allowed to enter into that. No other person will be allowed unless and until test facility management permission is sought for that. So moment of materials in and out of archiving should be properly regulated. Whatever material is once archived, it cannot be taken out. So it can be taken out for reference over there, but for short duration of time, a short period over there, and it should be with permission from the test facility management, and it should be recorded over there. And if the test facility out of business over there, all material should be transferred to sponsor archive. Obviously this case should not be discussed over here, but intention over here is very, because document says that if test facility goes out of business, all the materials whatever is available into the archiving should be transferred to the sponsor's archiving, or you can send it back to sponsor for that matter. Archival records and materials following should be archived for each study, like study plan, raw data, samples of test reference items, specimen, quality assurance, audited final report, quality assurance record, master schedule, all curriculum materials, CVs, everybody's CV should be there, job description, training records, maintenance and calibration record of apparatus over there. All this should be available into your facility over there. Now we have discussed 10 commandments, test facility management and organizing personnel, quality assurance program, what quality assurance program we should have over there, and what are the test facilities over there, suitable size, construction, all this we have discussed. Apparatus suitable intended purpose you must have. Material should be as per requirement over there. I have shared my experience over there. Whatever chemicals and reagent you are buying, they should be of high quality over there. Don't compromise on that. Test systems, yes, whatever test systems you are getting, before using test system for your experiment, you should certify them they are free from any health issues over there. So health monitoring program should be well in place in your facility over there. And whatever activity you perform in your facility should be with standard operating. No activity should be conducted or carried out in your facility without standard operating procedure. Jokingly, we used to say that even if at all rats are, breathing, there should be a standard operating procedure. This is extreme for that matter, but I'm just on lighter side. I'm just telling you over there. Performance of the study, you must how to perform the study, how to conduct the study. I've given in short explanation on this over there. And reporting of study results. Obviously, as I told you that GLP is only managerial system. GLP is not the science, and science, you and me have to do the science. Obviously, when you do the science, you must know how to interpret the results. Interpretation of results is very, very important. To write one sentence that your chemical is non-toxic or your chemical is non-carcinogenic, so obviously it requires a lot of efforts, lot of science, lot of skills for that matter, and you must be able to do that. So this is what is there. Reporting study results is very, very important aspect of GLP. And storage and retention of report, because you must know who has done that study at what point of time, and when GLP inspectors will come, they should be able to retrieve that particular study and check whether the study was conducted in your facility or not, whether you are existing over there or not, whether that rat was existing in that facility or not. Even rat, even when you have histopathology done, they will check against your tissue and against your slide. This is what GLP inspectors are going into details of inspection over there. Obviously, you need to be very particular in conducting GLP studies. That the reason GLP is very, very important, and you must follow OECD GLP principles over there. Thank you very much for this. Any question you can ask me for that matter. Any questions? Hello.
Hello, sir. This is Dr. Sachin.
Yeah, tell me, Sajjan.
Yeah. This is very nice presentation, sir.
Okay,
Thank you so much. So my question is that does it necessary to have a CCTV cameras in the animal rooms as per GLP?
Yeah, they said initially, but now they have not in room per se, but in lobby and all that. They said you must have that.
Okay.
Okay.
Yeah.
But
That means
for every activity, you should not be capturing for that. Initially, they thought of that, but now they have gone back to that, and now they said yes, you can have an entry point and in a clean corridor or dirty corridor over there.
Okay.
You must have cameras over there.
Okay. Fine.
Hello,
Thank you, sir. So one more question regarding the archival. So as per GLP, how many years we have to archive the our taste compound, or you can say the dry archival or the wet archival?
you need to find out from your test substance whether it is stable or not. But let me tell you that GLP asks us to archive for 9 years.
9 years.
9 years. 3 GLP inspection cycle, they say. So obviously
Okay.
3 GLP cycle, every 3 years they will come for inspection. So
Okay.
3 GLP cycles mean 3 into 3 is 9 years. So obviously you need to
Yeah.
keep 9 years. Whether it is
Okay.
stable or not stable, even you
Okay.
for all your test substance, you are supposed to keep for 9 years.
Okay. Fine. Okay. Thank you so much, sir. Thank you.
Thanks. Anybody has any other question?
Hello, sir.
Yeah.
Sir, there are like GLP guidelines from different
Can you
regulatory
just
bodies.
come on video?
I wanted to ask that there are GLP guidelines from different regulatory bodies like FDA, OECD, and WHO.
Yes.
How
Yes.
do they decide which GLP guidelines to follow in a GLP lab? And also, what is the basic difference?
Yeah, there are not much difference. There are some differences. Yes, obviously there are some differences, but that has to be decided by you. When you go for OECD GLP certification, so obviously you need to use OECD GLP only, and you can conduct FDA studies. You can conduct WHO studies or WHO vaccine studies over there, but the terminology what you are using, you should also use OECD as well as FDA and WHO for that matter. You cannot just forget about OECD and go about FDA. That will not be accepted by OECD or NGCMA for that matter. This is my experience I am sharing with you. Because GLP, because we are in India and whatever Indian inspectors they tell us, we need to follow that. So obviously, my experience goes that GLP inspector says, yes, you are OECD GLP compliance facility, so you must use OECD GLP and you add to that WHO or FDA, whatever you say. Is that right? Or Jyoti, you can
Yes.
correct me if at all.
Yes, sir. Thank you.
Yeah, definitely. So the sponsor actually needs to take a call where the particular test item is being submitted, which regulatory authority, like if it's US FDA, if it's going to be submitted, the study is going to be submitted to USA, then of course you need to follow FDA, right? And if it's UK, you have to follow MHRA. So it goes that way. It depends upon the sponsor, where the sponsor needs to file the study for marketing or research licensing. Are we clear?
Yes, sir. Thank you.
Thank you. Anybody has any question? If nobody has question, I have questions for you.
Great.
Okay. So there is a test for you for GLP knowledge. I am just testing all of you over here. Everyone has an opportunity to answer this question. Question consists of true and false, multiple choice or short answer over there. You must be able to. The question 1, what does NGCMA stands for?
That's a challenge question. Anybody can answer. Please.
Yeah,
National
anybody can
GLP
answer.
Compliance Monitoring Authority.
Sorry, who is answering? You
Sir,
just
this
say
is
your.
Josh.
Tell me.
National GLP Compliance Monitoring Authority.
Right.
Perfect.
Now, this is what National GLP Monitoring Authority. This is the answer over there. The second question, true or false? Question number 2. Training record should contain verification of training documentation. Whether true or false? Can somebody raise the
I think
hand
false.
and say that?
It's true, sir.
I think
Everybody
the one who
participate.
is
Everybody should
answering, just
participate.
speak up.
Sorry?
I would request the participants to say their name. If it's possible, they can switch on their video and then answer.
Yeah,
That'll
answer
be more
that.
interactive, right?
Yeah, more interactive. Yes.
Sir, it should be right, sir. I'm Karthik Panmare, sir.
Okay. It is true. Yes, you are correct.
That's great, Karthik.
That's good. That's great. The third question, dated historical copies of job descriptions of employees that have left the company can be permanently removed from the archiving?
I think it is
Please,
false.
please.
This is wrong, sir. This is false, sir.
It's false.
It is false. Yes,
False.
you are
That's
right. It is
right.
false. The multiple choice question now, we have choices given to you. The last date on which data is collected directly from the study is called study termination date, experimental end date, cancellation date, or experimental termination date. You have to select
Experimental
one.
termination.
Study termination date, sir.
Experimental
Okay,
termination date.
Experiment
you are
end date,
right.
sir.
You are right. You are right. Experimental termination date. Okay, very nice. Very good. Congratulations. So, second multiple choice question: who is responsible in assuring that there is a quality assurance unit? Who is responsible
Study director,
in
sir.
assuring there is a quality assurance unit?
TFM.
Principal investigator, test facility management, study director, or sponsor?
GFM.
TFM. TFM, sir.
Yes.
Study director.
TFM.
No,
Oh,
it's
no.
not study director. It's test
TFM.
facility
It is test
management.
facility management.
Facility
Yeah.
management.
Okay. Good. Thank you. Thank you. Now, true or false? Question number 6. Published literature can supplement SOPs.
Yes, true.
It's
True.
true. True.
True.
True,
True.
sir.
True.
No.
True? Sure?
Yes. Yes. Yes.
Sure?
Yes. Yeah.
Okay, I will give you answer. True.
Yeah. Yeah. It's true.
Okay. Okay. Now, last question over here for today's this thing. True or false? Question number. Quality assurance have the responsibility to write all SOPs.
False.
False. No.
False, sir.
False.
It's totally
False.
wrong.
False.
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 It's
False.
false. False.
Yeah.
Sure.
Yes, sir.
Yes.
Yes.
Yes,
False.
sir.
I will write false.
False.
Okay,
Yeah.
good.
Yeah.
Okay, thank you very much. Thanks a
Thank you.
lot for interaction. I appreciate your interactive participation in question answer. Let me tell you that we have also devised one question paper for you. It will be posted on our group over there, and you need to answer and submit it back to us. Okay.
Right.
So based on your assessment, certificate will be given to you. Right.
Okay.
Yeah,
Okay.
we have decided a certain competence level.
Yeah.
And when
because-
you answer those questions, accordingly, the certificates and the competence will be evaluated and the certificates will be delivered
Yeah.
on your mail.
So, if at all there are any questions, you can ask. Otherwise, I say thank you
I think
all.
there's one hand raised. There's
Okay,
one
okay.
hand
Let me
raised.
check. Let
Raghavendra.
me check. Let me check.
Hi, sir.
Tell me. Hello.
Yeah, hi, sir.
Hello.
So
hi.
nice meeting to you, sir. So regarding
thanks,
this
sir.
question is for the test item archive one.
Hmm.
So as per the guideline and your notice mentioned in the presentation also,
Okay.
it should be archived in the 9 years. It's the 3 cycles of the NGCM inspection.
Yes. Yes. Yes.
So some auditors are telling that one the stabilities, whatever you have told that one, whether the stability is there or not.
Yeah.
So what is the need for the 9 years for showing the expiries chemical storage in the facility?
Let me tell you that there is no written document available saying that it should be archived for 9 years.
Yes.
Okay. There is no document saying that. Yes, for document it is there, but not for test substratum. It depends. It's variable with our Indian inspectors. That's the reason I
Yeah.
say Indian inspections are different than other inspection which I have faced. I have faced German GLP. I have faced Netherlands GLP. I know that their inspectors, they look into what exactly you are doing and what is your SOP is telling about. You can also specify in your SOP what is that you want to do. Okay. There is no document available, but some GLP inspectors, they say 9 years over there. And
So we
it is
need
also,
we need to satisfy the inspectors or the regulations.
Yes. No.
principles.
Inspectors. You need to satisfy inspector, and that's the reason I am saying you need to keep for 9 years.
Because
Or,
the document says as per your national regulations, if you clearly read them. So
Yeah.
our national regulation goes to NGCMA. So as per them, it is recommended that you keep them for 3 cycles, right? So that
Yes.
is
Yeah, that
the
is what is
Because
there.
Because issue is coming in the 9 years for storage of the
I know that
test
most of the
items.
test items, they are not stable more than 2 years or 3 years, maximum 3 years. So obviously, keeping that type of test item in archiving has no meaning actually. Let me ask me, as a scientist, I can tell you about this. But
Yes.
GLP inspectors may not agree to that. What I
Okay.
say. What I say.
Because in case there is an inspection from a regulatory authority of another
Yeah.
country and you still have that test item, there are chances that can be analyzed for, critical issues in case
Critical
they
issues,
find
yeah.
something
Yeah.
really, difficult to get through. So in that case, it can be analyzed, right?
Yeah, if analyze also, it will not the rectification, the purity and the, because
Yeah,
stability
I
is
agree
Exactly.
with
over.
you.
It will not be.
I
But
agree
some
with
of the
you.
characteristics may be. Some of the
Yeah.
characteristics may be,
Yes, yes.
And after 9
Yeah, and
years, you are free to,
No, because
may
scientifically,
expose it
if
and
you
keep it.
look that, sorry, Jyoti, I'm just
No, it's
scientifically,
perfectly
this is
fine, sir.
not justified.
Yeah.
I am with you on this. Scientifically, it cannot be justified, but we can't justify our inspectors for that matter. Our inspectors are something different. Let me give you one simple example. One inspector, they asked the full form of OECD. Why I ask you NGCMA full form is only because of this. They asked OECD full form of OECD to one of the study director, and he was not able to answer. Then they pointed out one of the observation was your study director was not trained. So obviously we told them we have given this much training. They said we are not concerned about what training you have given. He is not able to answer my question. This was the answer over there. So this is what is happening with our inspectors. So obviously, one inspector asked whether you are conducting study as per 401. 401 is already abundant. You know that
Obsolete.
it is, yeah, absolute. It is absolute now. It is not in practice. But that study director was not able to answer that proper question, proper answer to GLP inspectors. It happens because although I say I am having 40 years of experience. If somebody asks me even at all at this point of time, what is the full form of OECD, I may not be able to tell you also, because it happens with everybody, every individual. It doesn't require that training for that matter, or it depends upon your brain capacity, mindset over there.
Yeah, thank you, sir. And one more question is having, sir.
Okay.
So that initial topic is the GLP. It is a definition. It is mentioned: planned, performed, monitored, and recorded, and
Yeah.
archived and reported. So
Yeah.
what is the sequence of line? Is the last one is the archived
yes,
and reported.
So basically, this sequence, I have not taken into consideration what sequence you are thinking about. Yes, it should be reported first and then archived. But GLP says that you must archive your complete report before sending it to sponsor.
Reported could be,
Yeah.
submitting to the regulatory authority. So in
Regulatory
that
authorities,
case,
you
this
can submit
archival
at any
has to
point
be.
time. Yeah.
Yeah. Yeah. So archival has to be done at a priority once
Yeah.
the study is completed,
You must
right? Once
you
the
must
study
define
director has
no,
signed the
no.
study report.
You must define archiving in your facility.
Exactly. Frequency. Yes.
Sequence
Frequency
over
has to be
there.
defined.
Yeah. You know, whether after completion of study, maybe short-term studies 7 days, maybe long-term studies 1 month, you can keep that in your SOP, because no activity should be performed without any standard operating procedures. So you must have that in place. It may vary facility to facility, doesn't matter, but you must have that standard operating procedure. So you can prove yourself to the GLP inspectors. Yes, we have this in our SOP and we work as per our SOP.
And it is recommended always to, do the archival as early as possible, because once the study report is signed, you don't have actually anything to do with it, right? Only the archiving would take its own time, right? Because it's a big study. So toxicological studies like 4-week study has a lot of specimens, lot of raw data, which has to be compiled. And, even the archival forms filling up takes a lot of time, so that takes its own time.
No, even, whatever, as I told you, use of computers over there, and you have data available on computer, that also has to be archived. Electronic
Yeah,
data
electronic
archiving
data.
has been taken into consideration, very important for this matter. So obviously electronic archiving has to be done. No data should be available on computers after the study is over. It should be archived within stipulated time period. You must decide what is the time frame for that matter.
Okay, sir. Thank you, sir.
Thanks a lot.
One
Where are
more
you from,
question
Raghavendra?
I am from Arazen Life Sciences, GVK Bio.
Okay. Wow, good. Thank you very much. Thanks for joining.
Thank you.
It's a great
Yeah,
pleasure.
thanks.
One more question from Piyush sir, actually.
Hello.
And before that, I request you everyone, please start your video for 1 minute. We will capture some photo for our memory. Yeah. Yes, Piyush sir.
Good evening, sir. This is Piyush Yadav from
Hello,
Vadodara Sun
Piyush.
Farm.
Yeah.
Yes, sir.
Nice
Sir,
to see
actually,
you.
yes, sir. Your presentation was actually very much informing, and even after working for years in GLP, we got to learn from you a lot. Sir, actually, my concern was regarding this. GLP says a lot about this HD and TFM and QAUs, but for the lab attendants, it's not saying a lot because most of the time the lab attendants are with the animals and the animal quarter, and they perform activities like animal checking, and in some facility in our also facility they are performing some pressure recording, and some activities they are performing, and even in the animal check crucial findings they can observe, like found dead or moribund when they are doing in the morning or the evening. So sir, is there any documentation, because I am not very much aware of the fact that they can report something to the
Very nice question asked, Piyush bhai. Let me tell you that it is very important. I have seen some study directors or some scientists, unless and until Babu is there, they cannot conduct their study. You got me? So obviously attendants are very important, and attendants can perform study without, they can handle animals much better way than your scientist.
Yes, sir.
This is what I have seen. Even I have worked with hundreds of veterinarians, so maybe more than that. But the attendants, they are handling animals superb, fabulous over there. But you must have training records for that. And lab attendant is your designation for that matter. For GLP, you don't require. It is study personnel only.
Exactly.
For you, is it
Because
study
even
personnel?
if they're recording
Yes.
something in the
Study,
raw data, they become
lab
a study
attendant,
person.
research assistant, research officer, they are not GLP designation. Let me tell you that. GLP is only study personnel and study director. These are only two things available over there. So there is no other lab attendant is not designation available in GLP.
Sir, sir, what will be the proper way of communicating the lab attendant to the study director? If he
You conduct study training program for them and write on their that there are training records you are supposed to do.
Yes, sir. They are having the training records. Yes,
training if at all training records are there, you must have job descriptions for them. You must have training records for them. And I really suggest every test facility once in a year for 1 or 2 months, whatever daily activities they are recording, you must write down in your job activity profile, job activity training records. Once in a year you do that. That will give you more impetus as far as their training is concerned. So what activity they are performing, what is that new things they are learning, write down in their job description, or write down in their training records for that matter. And this is very important, very important. Let me tell you that.
And if I can add, you need to have study that translated SOPs in case there are in case it's Maharashtra or some other state. Accordingly, there has to be translated SOPs to make them feel competent about what they are doing. They need to be trained on those translated SOPs in case they are not feasible or not okay with the English or which is normally taken. So translated SOPs will justify that they have been appropriately trained for that matter.
Yes. Okay. Thank you. Yes. Thank you, sir. Thank you, ma'am.
Thanks a lot. I hope I have justified your question.
No, sir.
Yeah.
Your knowledge is vast, and we are learning from you a lot, sir.
No, thank you very much. I know
That's right.
I have visited your facility when Dr. Marathe was there.
Yes, yes. Okay, thank you.
Okay.
Uh,
Thank you.
Sir, one more question. As you said, we cannot keep the two different species in one room, right?
Yeah.
So, can we keep these two different species if we have the IVCs, like individual ventilator
Yes,
cages?
yes. IVCs
right?
is different.
IVC.
IVCs are different. You can keep
Yeah.
IVC. One cage is independent room
Yes.
for that matter.
Exactly,
Obviously,
exactly.
IVCs systems are different over there.
Yeah, yeah, yeah.
You can
So
keep
in that
IVC.
case, we can. Yeah, yeah. Okay, okay,
Yes,
sir.
yes, you can keep that. No problem.
Okay, yeah. Thank you, sir.
Okay.
Sir, I
tell me.
have a small question that is GLP restricted
Who is
only
there?
for toxicology?
Can you just start your video?
I would just request everyone to just,
Yeah,
say your
unmute
name
and
from
use
your
your video.
yeah.
Yeah.
Your video, your
Yeah.
name, affiliation,
Yeah.
and then definitely you
Yeah.
can come up with your question.
Yes. Good evening,
Yeah,
sir. I am Prashant from Naipur, Mohali.
Wow, good.
I have a small doubt that is GLP only for toxicological laboratory or even for pharmacology, in vitro in vivo pharmacology laboratories?
No, it depends upon Jyoti. You will answer or I will answer?
Yes, sir. You start. I'll add on.
Okay. Basically, what type of studies, whatever regulatory submission is there, and you are performing some studies, it should be with GLP only. You cannot have non-GLP studies when you are submitting your reports to regulatory authorities. You need to find out from the sponsor whether you are submitting your
Exactly.
studies to regulatory authorities or not. Whether it is a pharmacology or toxicology.
Safety pharmacology study.
Yes,
like,
safety
for example.
pharmacology studies. Yes, there are certain studies which can be under GLP, but there are exploratory studies. You don't have to have GLP for that matter. I'm telling you that discovery program, you don't need your GLP for that matter.
The studies which are
Sir,
submitted in the IND, right?
But
So
anyway, preclinical data we submit to the regulatory.
yeah,
approval.
then it should be with GLP then in that case.
Okay, sir. So companies working on in vivo pharmacology, they need to have GLP certificate now.
Yeah, but Mohali, you already have, no?
Yes, sir. I had a small doubt.
Uh-huh.
Thank you, sir.
Yeah. You know
Sir,
Bhavin Kansara? Prashant?
Sir?
Bhavin Kansara, you know? Who is there in your facility only he is working as a, I don't remember now what he is working on. Anyway, no
sir,
problem. No problem.
sir.
No problem. Hello. Tell me.
Hello, sir. I'm Karthik.
Hello, Karthik. Where
Hi,
are
sir.
you?
Sir,
Okay.
I'm in a home.
Tell me.
Sir, I have one small doubt.
Okay.
Sir, so many colleges like Naipur, they don't have a GLP lab. Then they perform their research work on animals and all that, like in vitro, in vivo. Sir, they need necessary to perform their experiment according to GLP.
Not necessary, because they do most of the exploratory work, discovery work, because I am also on IIC committees of most of the big colleges in Pune over there. They don't need, but see, whatever GLP tells you, your animal should be healthy. You must understand that whether you do exploratory study or non-exploratory or discovery study, your animal should be healthy. You must take care of your animals. You need to have that facility at least with you, because I have seen in some of the colleges at the end of your college there will be small room where you keep your animals. No AC over there, no facility is there, food and feed is not there, water is not there. This type of situation should not be there. So at least take care of your animals, that's all.
And GLP basically comes into picture when there is regulatory submissions. I'm saying it again and again because that is the bottom line. When you need to have a regulatory submission, then only for a marketing licensing or for a research licensing, then you need to go for GLP, and that in that case it is mandatory.
And let me tell you that in American university, most of the universities are working as per GLP.
Exactly. So there's a hell lot of difference.
A lot of difference. Why we are not getting business from America only because of this? They do it at very cheaper rate and very up to date research. They get it done from universities over there, and they are giving regulatory submission from universities only. So obviously you need to understand the difference, and we must go for GLP in most of the universities or where you are having animal facilities at least. When you are having animal facilities, at least when we were constructing our animal facility way back in 20 years or 25 years before, there were no architects available in India to construct the animal facility over here. So obviously this was the situation. So nobody knows what is required for construction of animal facilities over here. This is a very sad state. At least it should start from the university. University should start, explaining what is the requirement of GLP. And
exactly.
Yeah. So this is my perception.
So much of a difference.
Yeah.
It's so much of a difference. Like if you put on paper, I think there'll be around 1,000 institutes and academic universities which are GLP certified in US. Yeah. And in India, I think everybody must be knowing we still have only
what is
50.
GLP?
So
they're talking about that.
There is a far more way to go ahead, a lot of scope, and I think for a good time.
We are maybe 100 years behind from them. Yeah.
No, sir, we don't. We won't. We don't. Worry. I think.
Thank you very much. It is 9 o'clock.
Yeah.
I think so. Time is.
So, can you take one more question?
Okay, tell me.
Yeah, is GLP concerned with the OECD guideline or some other guideline also comes under GLP norms?
It depends upon. You can go for FDA GLP. You can go for MHRA. MHRA will not come to India, but OECD comes to India, and we have OECD in India now. India has become member of OECD.
Okay.
You know that. Yeah. It's a very big achievement for government of India. In 2011, India became the member of OECD over there. So obviously this is additional, and we have a monitoring authority in India who monitors GLP facilities in India. Now we have more than 50 facilities available in India who those who are GLP compliant. GLP certified over there. Obviously, number is increasing. Their target is 100 facilities by 2025. But let us see. You must come out and see that you have GLP facility over there.
It'll be like adding economic value to the country. The more you are GLP, the more acceptable your data is.
That we are going to tell you about this in our GLP in India presentation. We are having GLP in India presentation. In that, we are going to tell you how you can apply, what is the foreign exchange we have saved for doing studies in India. All this is there on their site. You go to NGCMS site, you will find that available. So how many studies we have conducted and how much money we have saved, all this is available in a site over there, NGCMS site. Okay.
Okay, sir. Thank you.
Thank you very much. It's more than 2 hours for me.
And thank you so much, everyone, for being so interactive and joining this
session. Thanks a lot.
Yeah.
I hope you have questions. You can put it on group also. We have a group. In the group, you can put the question if at all you have that. Okay. Thank you very much. And we will look forward to see you on next Saturday. Next Saturday, Jyoti is joining me for presentation. So obviously, it will be a great day. Please join next Saturday.
See you all next Saturday.
Yes, see you on next Saturday. Thank you very much.
Stay safe.
Thank you very much. Thanks a lot.
Thank you, sir. Thank you.
Akash, you finished?
Thank you so much, sir.
Shakti Prasad sir wants to say something actually. Please.
Shakti?
Hello, good evening everyone. Actually, I want to say something which I supposed to say in the beginning of this webinar. actually. But there was lots of people was supposed to come join this webinar. That's why it was just remained. Okay, so I first of all I welcome. Okay, this should be done earlier actually, but I can welcome you all because this is the beginning of this webinar series too. And we are having the expert panel with us, and some of the experts are already with us. Dr. Deshmukh sir, Jyoti ma'am, Dr. Shraddha ma'am, and these all experts are associated with us, and some other dignitaries are Dr. V.K. Tiwari and Dr. Prahlad Wangikar and Dr. Thompson sir. They are also going to join us in next webinars, coming webinars. So and also we are actually we want to see each of you and also your colleagues or friends to join this webinar, as we want to promote or educate about GLP about all you guys. So our motto is that, and we will share the information booklet to WhatsApp group, as well as we are forwarding to your mails. But if you are not joined with our WhatsApp group, so please join as soon as possible. So we will provide all the information and coming webinars to you. So that's what I want to say, and I want to thank you everyone. And obviously to Akash also, because he is tirelessly working for this webinar. So that's all from me. Thank you very much.
Thanks to you, Shakti.
Thanks a lot. Thanks a lot.
Thank you so much.
Thanks a lot.
Good work on
the test assessment questionnaire will be shared to all of you within few minutes after this session.
Yeah.
So please do feel because that will be the assessment that will be considered for your assessment to provide you certificate.
Certificates will be given to everyone, but merit certification will be given based on your competency level.
Yes.
Hello, Shraddha, you want to say something?
No, I just wanted to compliment Shakti for making such a good those brochures and everything. It's a great work.
He has put a lot of effort.
A lot of
Thank you,
effort.
Shakti.
Clearly visible.
Beautiful.
Yeah.
She has done beautiful. Okay, thank you very much. Thanks a lot.
Yes, thank you.
Thank you so much.
Thanks a lot. Thanks a lot. And Shakti, one more thing, I forgot to take it before we start. Basically, you are supposed to introduce the authors.
Yes. Actually, you have you started. So next time,
please, next time you introduce me, introduce Jyoti.
Yes, definitely.
Introduce.
So I made
everybody's CV are there with you.
Yes.
So obviously you need to introduce them.
Sure.
Okay.
So I'm prepared all the things, but from
no problem. Next webinar you can do that. Okay.
Yes. Okay.
We are also learning every day. No problem.
Yes. Sure.
That's right.
Thank you.
Okay. Thank you so much. Good night, everyone.
Thanks a lot. Bye.
Good night, everybody.
Thanks.
Bye, Shraddha. Take care.
Bye.
Yeah. Bye, everyone.
Bye.